I just presented Rikard Landberg as Associate Professor from the Swedish University of Agriculture, but within 2 days or so he will start a new position at Chalmers University of Technology as Professor working with food and health. Congratulations to that, Rikard. Rikard's research has focused on health effects of cereal-based foods and the development of biomarkers for food intake and health effects. And today Rikard will talk about the role of cereal grain-related additives and ingredients in human health. I mean, we have touched this subject in many presentations, but now you will sum up there, it looks like.
Yes, please. I will try to do that. Thank you very much for the great opportunity to come here today and present both a summary and some results from recent studies in our laboratory. I was quick to change the title of my talk into cereal grain related constituents rather than additives, because of we know what an additive is and we know what a constituent is. So. Cereal. So whatsoever are the evidences we have for any health effects on cereals? I think that's important before starting discussing any particular constituent or ingredients on cereals. And of course, as scientists, we can go into different studies and look at the results of different individual studies, but we will find in this area a lot of conflicting results. So I would say that we have three general approaches to look into the comprehensiveness of this research and try to get an idea of what is the overall efficacy of different cereal components. One is the approved EU health claims, because there have been considerable amount of work on systematic evaluation of the scientific evidences behind health claims. We also have official recommendation by nutritional societies and also, of course, our national food agencies. And then, as scientists, we like to go into systematic reviews and meta-analysis also to try to ferret out the evidences. So when it comes to whole grain, I mean the grain as a whole, all the parts of the grain, there are consistent evidence for health beneficial effects based on epidemiology. And this is one of the meta-analyses conducted quite recently, where they went through 45 prospective cohort studies and 21 randomized controlled trials. And the results of this investigation shows that with increased intake of whole grains and cereal fiber, we might have a lower risk of type 2 diabetes, cardiovascular disease, and also weight gain, which is a main driver of the other conditions. And here they say might, and that's because these are observational data. This is not result of randomized controlled trials, so we might not need necessarily to know that this is cause-effect. And they also stated that the highest versus the lowest cereal fiber intake was associated with between 16 to 19% risk reduction of these conditions. Another thing that they came up with here, and which is also consistently shown in all these different types of evaluations, is the statement that we need more randomized controlled trials in this area, and we need randomized controlled trials of sufficient duration with well-characterized material. We participated in a large European study to look into the role of whole grains in relation to colorectal cancer, because colorectal cancer is one of the other areas where whole grains have been shown consistently to have a beneficial effect. But in this study, we used for the first time an objective measure of whole grain intake. So we have developed a group of biomarkers that we can measure in the blood as a reflector of the whole grain intake. And in this population, it was a large European population study. We looked at the association between these biomarkers in the blood and the incidence of developing colorectal cancer. And overall, we didn't find any association for colorectal cancer overall, but colon and rectal cancer are somewhat different diseases. So when we looked into subgroups of cancers, we see in Scandinavia, where the whole grain intake is very high, we have a protective association with colon cancer. And if we look also into proximal colon cancer, we don't see anything, but in the distal part of the colon, there is an inverse association all across Europe. And there is also a gradient that is following the gradient of cereal consumption. So our conclusion of this study is that we find that cereal fiber seems to be very important for colorectal cancers, as studies have shown fiber in general, but not many studies have looked into cereal fiber specifically. At the time when we were doing this study, there was a large meta-analysis published, and that meta-analysis showed very clearly that cereal fiber is the fiber that drives the protection for colon cancer, and particularly for distal colon cancer. So that was very nice to see. So, and I can say that the picture is similar for type 2 diabetes. We have, there are 15-20 studies on type 2 diabetes showing consistent inverse association. So with that in mind, you would perhaps regard that EFSA would come up with a beneficial statement for whole grains, but that was not the case. EFSA said that whole grain is not sufficiently characterized in relation to any claimed effect, and those claimed effects that were at investigations were these ones. So it was not proven that whole grain as a sort of concept has any beneficial effects into these different outcomes. And the reason for that is that EFSA says that whole grain has been poorly defined and is differently defined. There are different definitions used for whole grain. And we also know that whole grains as such are very different. I mean, the cereals are different, although they have also commonalities. In US, FDA has been less strict, so there has been a health claim available in United States since 1999, relying on epidemiological findings, allowing companies to say this on food packages on whole grain in relation to heart disease and certain cancers. And there is also more recently one very odd kind of statement. You can wonder why it's possible even to do such a statement, saying that whole grains may reduce risk of type 2 diabetes, although the FDA has concluded that there is very limited scientific evidence for this claim. This is watery. Despite the statements that we can't do on products, industry cannot claim health effects of whole grains, but our governmental authorities, they can provide dietary guidelines to promote whole grain intake. So in Sweden, for instance, National Food Agency says to the population, whenever consuming grains, it should be in the form of whole grains when you consume grains. And that's basically based on these epidemiological findings. So it's a little bit contradictory here. I think that companies cannot advertise health effects, but authorities can make general guidelines to population in this regard. So if we now want to understand what is it in whole grain that gives these beneficial effects that might be around? We have heard many times today that the grains, the different parts of the grain, and in whole grain we have all the parts of the grain present. We have the bran, we have the germ, we have the starchy endosperm. And of course, when it comes to bioactive components, we have most of them sitting in the outer parts of the grain and also in the germ. And also the same for the dietary fiber, which is mainly located in the outer part, but also in the endosperm, of course. And just to pinpoint that grains are different, I mean, this is just some figures on dietary fiber, is USDA data. So you can see here for rye, for instance, where we have almost 20% of dietary fiber, whereas in rice we have 4%. So they are very different, and this is also comes to different fractions of grains, where we have huge amount of fiber in the bran, whereas in the starchy endosperm the content is lower. The problem is when doing epidemiology, we cannot do epidemiology on different fractions, and we can hardly do epidemiology with different grains. So in epidemiology we can only investigate total whole grain, because the tools we have to estimate intake is not precise enough to study these differences, although we have seen today that there are huge differences in the fiber composition and also effect on microbiota and so on, and this is a problem. We also have a lot of bioactive compounds in grains, and those also vary in different grains and different parts of the grains and so on. And I can see right now that there are no conclusive evidence whatsoever for bioactive compounds related to health from grain. Fiber has been around and discussed a lot here and in other places, and fortunately we have now uniform definition of dietary fiber, which says that it's an eatable carbohydrate polymer naturally occurring in the food as consumed, and also polymers that have been isolated from raw material or even synthesized polymers. And this has been around, of course, for the evaluation of health effects. And we have heard also that fiber is not just fiber. We have large diversity of fiber, and this will have also influence the role it has on health, of course. So we have the natural occurring fiber, which comes from the different parts of the grain, which also contains the associated bioactive compounds. We have isolated, and we have also synthesized fiber. And as heard earlier, you can divide them largely into soluble, insoluble, or fermentable, or unfermentable. And what about the evidences for dietary fiber from cereal grain in relation to these diseases? I would say it follows very much the evidence from whole grain. It's just that because they are very highly correlated, and this reflects the problems also to actually study individual components, because there are such a big correlations between these components, so it's hardly possible. But I just picked this study published in British Medical Journal, which is very interesting, because here they divide fiber into fiber from different sources, and that's a very rough way of dividing fiber, different types of fiber qualities in relation to disease. And again, you can see that fibers from cereals is associated with lower risk of cardiovascular disease. It's also associated with lower risk of developing coronary heart disease. And actually, it's much stronger, the protective association, than for fiber from fruits and vegetables. So again, it seems that cereal fiber is a main driver of beneficial effects of fiber, if there are any. What does EFSA say then against dietary fiber? Well, EFSA comes again and say that the dietary fiber is not sufficiently characterized in relation to any claimed effects. So we cannot use dietary fiber as such to do any claims on the products in Europe. However, in contrast to whole grains, we can analyze fiber very specifically, and we can define what is the dietary fiber component analyzed. And that means we can characterize dietary fiber and therefore also study it in relation to disease or to physiological function. And therefore, there are claims for specific dietary fiber components which can be used in Europe. And this is one of the perhaps most important ones, and it's beta-glucans. What is interesting is also to know that, of course, the different types of variety of beta-glucan will have very large differences in its physiochemical properties. It will be due to the structure of the beta-glucan, and it will be due to the molecular weight distribution of beta-glucan, and that will give certainly very large differences in physiological effect. Another important feature, which is very particularly shown for beta-glucan, and it's underlooked for in other fibers and other components, is the effect of processing. These are different food products based on the same raw material, and you can see here these are the molecular weight distribution in porridges, extruded oats, and breakfast cereals, and you can see that there are high molecular weight beta-glucans in these products, whereas when you produce a bread, you degrade the beta-glucan molecular weight. And that has detrimental impact for the function of beta-glucan as cholesterol-lowering agencies, for instance. This was the first study, really large, well-designed study to show that, where they show that you have a lowering of cholesterol with a molecular weight. So you need a certain size of the molecules to get the cholesterol lowering. And there is a very tight relation between viscosity and molecular weight. So physiochemical properties of the dietary fiber is very important for its effect, and in this case for cholesterol reduction. However, we have two claims on beta-glucans and cholesterol reduction in Europe, but these claims are ignoring this fact. So there was a scientific opinion by the experts in EFSA suggesting that it should be added minimally processed in this claim, but that was abolished. And then it's as a scientist, you start to think about when politics goes into decisions, and you wonder why. I mean, because simply you won't have the similar effect on cholesterol reduction if you start to process this material. So if you pour whatever 3 grams which is degraded, you won't see any cholesterol reduction, but still you can make the claim. So that was cholesterol reduction attributed to dietary fiber. What about other functions of dietary fiber where we have health claims? This is one. cereal fiber and bowel function. This has been very well established for different types of cereal fibers, and for wheat you can even calculate how much more fecal bulk you will get per gram of wheat fiber that you eat. Very useful information, and I don't think the companies can make that much out of it actually. But it's possible, we know what is going on there. When it comes to fiber and also reduced glucose rise, we have certain claims allowing us to say that if we have a certain amount of these fibers, we will affect the postprandial glucose responses beneficially. And for instance, arabinoxylans produced from wheat endosperm, beta-glucans again from oats and barley, and also resistant starch. So, but looking through this, we have rather few indications where we have sufficient evidence to say that cereal constituents are affecting our health. And this is with certain proof that we can allow health claims, at least in Europe. And what studies come up with is also that there is very often an inconsistency with epidemiology, that typically observational studies show beneficial associations, but they are very difficult to replicate in randomized controlled trials. And part of the problem is the characterization problem. People are not characterizing the food material that they are testing in these studies very well. And that's why food scientists are needed so desperately in these kind of multidisciplinary studies where you study effects of health effects of foods. And it's also very difficult to attribute effect to specific components in food because we have such a high correlation between different components in food. So more research is needed, and I will give you some examples of 2 studies where we have evaluated metabolic effects of prebiotic fiber and also fermented rye bran in 2 different studies. In this study, we wanted to evaluate, we have done a number of studies in our lab investigating the role of rye when it comes to appetite and metabolic effects. And we have shown that rye is more satiating than wheat. We have also shown that you have beneficial postprandial insulin responses if you consume rye. But there has been a lot of fuss about microbiota lately and about the role of microbiota also for appetite signaling. So fermentation of dietary fiber producing metabolites that might affect appetite signaling. So in this study we wanted to investigate the specific role of fermentation by means of increasing satiety in people and also giving beneficial postprandial insulin and glucose responses. So what we did was that we basically took a rye porridge and we took away 20 grams of the rye and added inulin, which is an easily fermentable fiber, together with gluten, because gluten was the only protein that had sufficient pleasant eating properties, basically. So that's why we selected gluten. So we mixed gluten and inulin in different ratios. We fed people this, and then we measured appetite. We measured also glucose and hormonal responses. So we had 21 subjects, and all the subjects had the different diets in different orders, so it's random order. So 40 grams of rye. We had also plain rye porridge with 50 grams and 40 grams as controls, and then we have added inulin and gluten in different combinations, and we also have a refined wheat as a control. And then we measure the appetite. And the gold standard for measuring appetite is actually to ask the person, how hungry, how full are you right now? And then they have to answer on a scale from 0 up to 100. And it sounds very unreliable, but in fact it's very reliable within individual. And since all these comparisons are made within individuals, we think it's reliable. So when they came in the morning, they were quite hungry, so they were not full. And then they had a porridge, and then you can see fullness increases. And it increases significantly more for the rye porridges compared to the refined wheat. Then they had lunch, and then they got full again, but then there is no difference. And what we suspected was then that those who had a lot of inulin, they will have huge fermentation going on, they will feel more full after when fermentation kicks in. But it seems that we don't see that here. And then it's the question, why don't we see that here? First of all, it could be that fermentation was not that efficient, but we measure fermentation by a breath test. So we analyze hydrogen and methane in the breath of these people, and you can see that there is a very nice dose-response relation. With increased amount of fiber with added inulin, we have more fermentation going on. I should also say in these studies we didn't see any beneficial effect on postprandial insulin, which we have seen in all other previous studies on rye, but those studies were on rye bread, and this is rye porridge, and that also shows that the processing, the matrix, has also probably impact on the results that we see. However, what was very interesting was that we saw after lunch, when we measured the glucose concentration in the blood, we see that the glucose goes down more rapidly in the blood among those who consume the rye porridges compared to those who consume the wheat. So we have beneficial effect on secondary meal effect on glucose in this study. And we thought that this might then be due to the short-chain fatty acids, so we have developed a new method which is more precise, robust, and also more sensitive for analysis of short-chain fatty acids, butyrate, acetate, and propionate, and also some other fatty acids in blood. And what we could see was that we had the highest concentration in those who had most inulin, and that was kind of expected. But we didn't find any correlation between short-chain fatty acids and the responses in glucose, and that surprised us. We would have expected, if fermentation and these metabolites would be important for glucose regulation or appetite, we would see a correlation, but there is no correlation. We also measured metabolomics, so we used NMR to analyze a large number of metabolites because we wanted to see what metabolic pathways and metabolites are shifted and can be related to our differences in appetite, and also this second meal effect on glucose. But also there we analyzed a large number of metabolites, but actually we see no changes which can be related, which is a little bit surprising. So our findings was that porridge, rye, whatsoever, rye with inulin or gluten, you get more full than compared to wheat, and that was not a big surprise, but the surprise was that we don't have any effect attributed to the inulin. Why don't we see any effect of insulin as we have done in other studies? Perhaps it's explained by microstructure, because porridge is completely different structure if we look in the microscope than other rye products. Gut fermentation increased, but we found no relation between secondary meal effects and short-chain fatty acids, and it appears that rye, rather than the inulin and protein, affects the second meal effect. And actually this study left us with more questions than answers. We still don't know what is the role of fermentation and short-chain fatty acids in appetite regulation. Probably there are other mechanisms that are involved. We are looking into that in more detail now. Then I will show you some other results. This is another study we have done. The previous study was with inulin and gluten. In this case we have investigated the role of fermented rye bran. By adding fermented rye bran to products, we wanted to see if we can reduce the bacterial load of Helicobacter pylori, which is the stomach ulcer bacteria in the stomach. And we also wanted to evaluate for the first time what is the metabolic effect of such a high rye consumption when a large number of people are eating rye for 12 weeks. And we went for doing a study in China, because also Chinese people, they are not used to eat rye. The problem in Nordic countries when we do these kind of studies is that they habitually eat a lot of rye. So we did this study in China, a 12-week intervention, together with Fudan University in Shanghai, and also together with two companies and collaborators at Umeå University. And the reason for why do we believe that cereals and fermented rye bran has anything to do with Helicobacter pylori, it sounds like a very crazy idea. But the reason for that is that it has been shown that cereal fiber is associated with lower incidence of gastric cancer. And gastric cancer is a cancer form which is very prevalent in the Asian countries. And it's highly affected. The risk of getting gastric cancer is highly affected and modified by the presence of stomach ulcer and Helicobacter pylori. And Helicobacter pylori prevalence is very high in China. So then there has been a lot of model experiments conducted with different food products, isolates from supernatants from different food products, screened for the effect on adhesion of Helicobacter pylori in the stomach, going on in different labs. And that has proven that a specific strain of lactic acid bacteria combined with rye bran seem to have a beneficial effect on adhesion, preventing the bacteria to adhere to the gastric mucosa. So that's the background for why we wanted to try to prove it also in a clinical study. And there have been also studies, pilot studies conducted in China with some positive results. So, and I was very interested to look at metabolic responses after such a long time on rye. So we designed a 12-week intervention. So we gave people rye bran, fermented rye bran in different products. So we screened 569 people, and we selected people who had Helicobacter pylori. And how do we know? We are using a breath test. So we give them urea, label urea, and if you have Helicobacter pylori, they will break it because they have urease activity. So they break the urea, and you get carbon dioxide, label carbon dioxide in your breath, and that's in proportion with the bacterial load. So the more labeled carbon dioxide, the more bacteria you have. Not necessarily the higher severity of the disease, but at least bacterial load you have higher. So we gave them fermented rye, and we measured breath test, we took blood samples, and we also took fecal samples, because again here microbiota might be interesting to follow as well. And we did that for 12 weeks. And the products they got was a black box. This is the fermented rye bran with a specific bacteria. It's a patented stuff that was mixed in 25% into crisp bread and also to these kind of puffs. A problem is of course to introduce these kind of products to a population who is not used to eat it, and that will show also. We had some problems with that actually when evaluating the data. Even a Swede would have difficulties on eating this actually, because it's very bitter and it tastes sour actually. So it was a challenge. But these people had quite a lot actually. So they should have 4 slices of crisp bread, almost 100 grams of these puffs, and by doing so they will get, in the rye group, they get 37 grams of dietary fiber from these products, so it's quite a lot of dietary fiber they get. And the match control products, they get 12 grams. And this was the part of the investigator team. They put up a research center. During 2 days they sampled 200 people. It was quite impressive to see actually. Here you can see the products. That was a nightmare. It took 1 year to ship products from Sweden to China with all the paperwork, but we managed at least. They took the blood samples in the study center, handled blood samples. And this is how it looks like. So we included 262 out of these 562, randomized 180. We had 90 people in each group, and after 12 weeks, 80 were still completing. So it was not a big dropout during the study. We also had follow-up measures 3 months later, just to see if any changes, if they would be gone after 3 months. Disappointingly, we didn't see any effect on the breath test, so we don't see any whatsoever effect in our primary outcome, which was disappointing. But what we see, what is very interesting, is that obese people, because half of the people were randomized obese in the 2 groups, and there are very large differences in the bacterial load among obese and non-obese people. And there are also some indication for interactions of some of the effects. So obesity, which we know from the literature also, they have there are links between certain gut microbiota and obesity, and there might also be links with Helicobacter pylori with obesity. So this is something we are now want to look more into in detail. When we measure the blood parameters for metabolic effects, we don't see anything except for cholesterol. And this is actually the first randomized study where we can prove on the beneficial effect on cholesterol reduction. So we reduce LDL cholesterol and we reduce total cholesterol by feeding these people rye. And that has been shown in pilot study, unpublished studies in Uppsala before, but this is the largest study to actually prove that. And actually the effect is rather similar as you would see for beta-glucan in oats. So that's something also that we want to continue with. because this is interesting. This is very interesting if you can obtain a cholesterol reduction with rye also. So what we find so far, no significant effect in the primary outcome measurements, although we are still waiting for microbiota to be analyzed as well, which will be interesting. We see the effect on cholesterol, and also I should say that compliance was an issue in this study. We measured this alkyl resonance, this biomarker of whole grain wheat and rye intake, and we see that there is a huge number of people who don't eat what they should eat. So we will reanalyze the data, also taking that into account, eliminating those people who are not compliant. Appetite, we will also look into appetite data here. So, how to sum up then? The role of cereal constituents and ingredients for health, some conclusions. We see that there are proven beneficial physiological effects, at least, for several specific dietary fiber components from cereals. What it means for health, that's another thing, but it's a beneficial physiological effects that have been observed, which health claims are available for. Evidence for bioactive compounds are completely inconclusive, I would say. So we don't know what is the role of bioactive compounds in cereals when it comes to health. I think more emphasis, as many other authors have stressed, is also that we should put more emphasis on the role of food processing. We have done other studies I didn't talk about, for instance with sourdough, showing complete adverse effects on postprandial glucose and insulin responses with sourdough, for instance. So more emphasis on the role of food processing for health effects of grains. Also, more randomized control trials are needed. These should be long-term studies, also using well-characterized materials. And then we should take the advantage now of omics technologies, that we can do high-throughput omics in large sample sets, both for randomized control trials, but also observational studies, in order to understand any mechanisms that underline these physiological effects that are observed. I would like to thank you, and I also would like to thank my collaborators and also my group in Uppsala. Thank you very much for your attention.
Well, Rikard, that was a rapid presentation on what we know about cereal and health. You're asking for large intervention studies.
Mm-hmm.
Do you think that is possible to do? Many of those?
Yes, I think so, and I think in the Nordic countries we have a huge number of very good prospective cohort studies, and that is something that has been started also. New large prospective cohort studies where thousands of people are sampled, they are characterized with regards to health and diet and so on. I think this is a splendid opportunity to ferret out and go back to these people and invite them for targeted dietary intervention studies. So if you are interested in men with metabolic syndrome, recruit people from these prospective cohort studies where they have already been characterized, and a lot of samples have been analyzed for a large number of measurements. That's an ethical issue. You can't go back to people and ask them in these kind of studies, but for foods you can, because we want to say to them that we want to investigate something which will benefit your health. We are not looking for risk variants of cancer or something like that. We will ask them, do you want to participate in an intervention that might help you to get a better health, just by means of foods. So I think it's possible to do that.
Because that's otherwise, it's very costly to do those studies. I mean, one human study of that size is like 10-15 million crowns, so you really must be sure that you get some results out of it before you start this type of studies.
I think it's very costly to do the other types of studies that is doing. I mean, small studies, underpowered studies in very specific groups, it also costs a lot of money. It's several PhD student projects. I mean, and what doesn't that cost in this country particularly? I mean, and no results. So that's.
Okay. We have a question there, please.
Hubert de Leiker, EFSA. I'm not going to talk about health claims, but I'd like to follow up on your comment that these studies are very expensive. I find this actually astonishing in the environment that we live, that we have 10 billion set aside for research funding in the medicines area, which is solving problems after everything has gone wrong. And we're still in a humble environment here where we say when we talk about prevention, we struggle to get research money to do exactly as you propose.
Yeah, thank you for that comment. I agree completely. Okay, I think we have to stop here. Thank you again, Rikard.
Thank you.